It is my pleasure to present to you the marking tips or the way Marking Biology Practical 2015 Paper 3 was marked. So I will begin by highlighting what was involved. In the first photograph there, you can see different animals. We have the eagle, the fish, earthworm, tortoise, octopus, starfish, spider, and frog. Next, we hand for question 2 the cervical, the thoracic, and the lumbar vertebrae. You can see F, G, and H. Then question 3 was about Fundi tests, and you can see those colors there indicate that this practical was testing presence of starch and presence of reducing sugar, as you can see the blue black color. Question 1 was, using the pictures of the animals provided below, complete the construction of the dichotomous key by filling the blank spaces. So those are the photographs of the animals which you are supposed to use, and use the dichotomous key. All right. So that's the key you are using. It's up to number 9. Number 1 to 9 steps. Question 2, below are the pictures of 3 mammalian vertebrae, F, G, and H. We will see what the questions were all about. So identify the type of vertebrae, F, G, and H. Label 5 parts of the vertebrae, labeled H. Name the articular facets, labeled K and L. How does each of the parts of the vertebra enable a mammalian skeleton to carry out functions? Question 3, you had been provided with 250 ml beaker, 4 test tubes, solutions labeled D and E, iodine, and Benedict's solution. Have filled the beaker with the hot water provided to create a hot bath. Label the 4 test tubes as follows. Test tube 1, D plus iodine. Test tube 2, D plus E plus iodine. Test tube 3, D plus Benedict's solution. And test tube 4, D plus E plus Benedict's solution. You are putting 1 cubic centimeters of solution D in each of the 4 test tubes. To the D plus iodine test tube, add 1 drop of iodine solution and shake to mix. To the D plus E plus iodine test tube, add 1 cubic centimeters of E and 2 drops of iodine solution. To the D plus Benedict solution test tube, add 1 cubic centimeter of Benedict solution and shake to mix. And to the D plus E plus Benedict solution test tube, add 1 cubic centimeters of solution E and 1 cubic centimeters of Benedict solution, shake to mix. Observe the changes in each of the 4 test tubes. Put all the 4 test tubes in hot water bath and observe carefully for about 5 minutes. That was what the question was testing. And there was the table which you are supposed to fill. Number 1, 2, 3, 4, numbers of the test tube, what is there, D plus iodine, D plus E plus iodine, D plus Benedict solution, D plus E plus Benedict solution. Then you have the observation and the conclusion. What was the law of each of the following in the experiment? Solution E, hot water bath. In B, give the identity of E in human beings. And C, explain the observations made on the reagents tested with Benedict's solution. Have you ever wondered how scientists can quickly identify an unfamiliar plant or animal in the world? What if I told you there is a simple step-by-step method to do it without being an expert? By the end of this video, in question 1, you'll be able to confidently use a dichotomous key to identify organisms in nature, whether it's for your next biology class, a field trip, or just for fun in your backyard. Even if you have never used a dichotomous key before, don't worry. I walk you through each step, breaking it down with simple examples that you can follow along. Let's get started. But before we get started, I would like us to know what is a dichotomous key. A dichotomous key is a tool used for identifying organisms or objects based on a series of choices that lead to the correct name or classification. It consists of a sequence of paired statements or questions, where each pair offers two contrasting options, often yes or no, or true or false. By making a choice at each step, users progressively narrow down to the possibilities until they arrive at the correct identification. Dichotomous keys are widely used in biology to identify plants, animals, fungi, and other organisms. They are structured in hierarchical way, with each step further refining the identification process by focusing on specific characteristics such as size, shape, color, or behavior. So guys, that is the meaning of dichotomous key, or how you apply the dichotomous key. So let's dive into the way this examination was marked. So there, I'll briefly let you write down what is a dichotomous key. So it is a tool used for identifying organisms, as I've said, or objects, based on a series of choices That leads to the correct name or classification. And I've already delved on that. So let's dive into the real things which brought us here. So complete the dichotomous key. And here you are getting 13 marks. And you have number 1A, animals with a backbone. You go to 2. Animals without a backbone, go to 5. Animals with wings, you see, you have identified eagle. Animals without wings, go to 3. In 3, animals which live in water all the time, fish is identified. Animals which live in water sometime, you go to 4. 4A, animals with scales, you have identified tortoise. Animals without scales, frog is identified. 5, remember where we were told to go to 5 in 1B. Animals with legs, you go to 6. And B, animals without legs, go to 7. So we go to 6. Animals with 6 legs, butterfly is identified. B, animals with 8 legs, spider is identified. Number 7, animals with a shell, snail, identified. B, animals without a shell, go to 8. Animals with a jelly-like body, go to 9. And 8B, animals without a jelly-like body is the starfish. Remember in 7B we had been told to go to 8, and we have identified the starfish. 9, animals with a segmented body are the worm. Animals without a segmented body, octopus. Question 1. These are the precautions. Reject if in the key you don't have the words go to, or if go to is missing, you don't mark. Also reject if identities are wrongly spelled. Again, if identities are written in plurals, because they are not in plurals here, steps are inserted on the dotted line. And finally, if the guiding step is wrong, all subsequent steps are wrong. That is the way you marked question 1. So, subsequent steps. 1B, 5A, 6B, 7B, 8A, 8B, 9A. 2B, 3A, 3B, 4A, 4B. Correctly repeated answers, one on dotted line and one on the space, give the first and equivalent and tick the one on the space. This question is testing construction of dichotomous key and not use of a dichotomous key. So no steps are required in this question. Steps are required when using the key and not construction. Let's go to question 2. The types of vertebrae are cervical, F, G, thoracic, H, lumbar. You label 5 parts on the vertebrae H, and the marking was done clockwise from the neurospine first 5. Other parts are equivalent. Labyrinths should not touch the part labeled. Labyrinths should not cross. Fine. Did you know the health of your entire body depends on just 5 vertebrae in your lower back? Discover how these powerhouses hold you up every day. By the end of this section of the video, you will know exactly how the lumbar vertebrae support your body, protect your spinal cord, and help you move, all in a way that can improve your posture and overall well-being. Whether you are a student, a health enthusiast, or just curious about your body, we will break down every part of the lumbar vertebra in a simple and engaging way. From the vertebral body to the spinous process, you see why each part matters and how they all work together to keep you standing tall. So here is a detailed drawing of the lumbar vertebrae with clearly labeled parts, including the vertebral body, vertebral column, spinous process, transverse process, and articular processes. I hope it will help you with your understanding. Remember, guys, there was a place where we were labeling on the vertebrae, but unfortunately I misplaced that diagram. So I used digital access to this, to what you are going to see. So you can see vertebral body, vertebral foramen, all right. And there are some languages there, vertebra porcine, vertebra foramen. So far as, okay, spinous, transverse process. I think it is spinous process. I think it is, this uproot was mixed with other languages. And here we have transverse process. All right. So we were supposed to label on the vertebrae, which was given in the question. Drawing and labeling of the 5 parts of the lumbar vertebra. A lumbar vertebra is part of the lower back, known for being larger and more robust to support the weight of the body. While I cannot draw directly here, I will explain how you can visualize and label 5 parts for a lumbar vertebrae. We have the vertebrae body. The large solid portion at the front of the vertebrae. We have the vertebral foramen, the hole in the center where the spinal cord passes through. Then we have spinous process, the bony projection at the back, which can be felt when touching your spine. Spinous process. Transverse process is the 2 bony projections on the sides of the vertebrae. And articular processes, the small flat surfaces that connect with the vertebrae above and below. From the drawing, you are supposed to sketch, if you are drawing yourself, you are supposed to sketch a rounded vertebral body in front with a central hole, and then and the spinous process extending backward, transverse process to the sides, and the small articular processes for connections. In my future videos on the other years, because what I've done is I've tried to cover the practicals from 2001 to 2015, I'm going now to go back downwards up to when the KCSE or 844 was started, and we will see actual drawings in that. You know, as the technology improves, the examination council has changed the tactics So instead of you drawing directly, they give you a photograph. Okay. We will see in the other papers. You will see in the other papers in my coming videos. Name the articular facets labeled K and L. You have seen is tubercular facet. All right. And L is capitular demifacet. They are there in those photographs. How the parts of the vertebra enable a mammalian skeleton to carry its function. Wrong neurospine for attachment of abdominal muscles. Neural arc to protect the spinal cord. Extended transverse forces for attachment of abdominal muscles. Neural canal for passage of spinal cord. Facets for articulation with other vertebrae. Tubercular and capitular facets for articulation with ribs. Large centrum to support the body weight. Vertebratal canals for passage of blood vessels. Nerves and vertebral arteries. Reject veins and capillaries. And then we have the metapophysis for attachment of muscles. So, looking at the digital world, I was able also to research. Remember, when I give these videos, I'm also researching on whether we as examiners exhausted everything. So if there is something which we left, I'm putting it back so that I give you the complete information about the video. So we have the vertebral body. The function, the vertebral body is designed to bear weight and withstand compression. You see now, this answer is a bit deeper. It has depth. How it helps, it provides support for the body structure and is crucial in maintaining upright posture by carrying the bulk of the body weight. Vertebral foramen. Function is protects the spinal cord, which runs through this canal. How it helps, safeguards the delicate spinal cord from injury while allowing nerve signals to travel between the brain and the rest of the body. Spinous process. Function provides attachment for muscles and ligaments. How it helps, it facilitates movement and stability of the vertebral column by allowing muscles to attach and pull during bending or twisting. Transverse process. Function serves as attachment points for muscles and ligaments and provide leverage for movement. How it helps? These projections assist in the lateral movement and the rotation of the torso and contribute to the stability of the vertebral column. Articular processes. Function form joints between adjacent vertebrae, allowing for controlled movement and flexibility. How it helps? They help limit excessive motion, protecting the spinal cord and allowing flexibility such as bending and twisting without compromising spinal stability. These parts of the lumbar vertebrae work together to support, protect, and enable movement, making them integral to the skeletal system function. So we are now in question 3, and we ask ourselves this. Have you ever wondered how scientists can tell what's really inside the food you eat with just a few simple tests? What if you could do the same in your own kitchen? By the end of this video, you will be able to, this section of the video, you will be able to confidently test for starch, proteins, sugars, and fats in everyday foods, whether it is from biology experiment or just to satisfy your curiosity. But you are not supposed, don't worry. But don't worry if you ever tried this test before. I'll show you step by step how to carry out these simple food tests using iodine and the Benedict solution and more. You'll discover the hidden nutrients in common foods like bread, milk, and even cooking oil. Question 3. Observations and conclusions for each of the 4 test tubes. If the table is blank, reject everything. That's the way we looked at it. So, question 3, we had mentioned about the test tube and the number. So let's just go to observations, and you find that in number 1, blue black, starch present. In number 2, the color turns blue, colorless, turns colorless or blue, starch absent, and so on. Or color fades, starch decreases or traces of starch. Number 3 remains blue, reducing sugar not present. And in test tube 4, turns green, yellow, orange, brown, or red brown or reddish brown, reducing sugars present. But if it turns only green, you conclude that there are traces of reducing sugar. So here is an explanation of each test, including the observations and conclusions. Starch plus iodine solution. Observation, a blue-black color appears. Starch, D was a starch. Conclusion, the blue-black color indicates the presence of starch. Iodine reacts with starch, forming this characteristic color. Number 2, or test tube 2, starch plus salivary amylase plus iodine solution. So D was a starch, E was salivary amylase. Observation, non-blue-black color remains yellow-brown, the color of iodine, after some time. Conclusion, the absence of blue-black color indicates that starch has been broken down by the salivary amylase into simpler sugars. Salivary amylase breaks down starch into maltose, so iodine no longer detects starch. Number test tube 3, starch plus Benedict solution. Because D was starch. Observation, no color change, or it remains blue after heating. Conclusion, there is no reducing sugar present. Benedict solution tests for presence of reducing sugars like glucose or maltose. But starch is a monosaccharide and it does not react with Benedict's solution. Test tube 4, starch plus salivary amylase plus Benedict solution. Observation, after heating, the solution turns from blue to green, yellow, or brick red, depending on the amount of reducing sugar present. Conclusion, the color change indicates the presence of reducing sugars, maltose, which are the result of starch being broken down by salivary amylase. The amylase hydrolyzes starch into maltose, which reacts with Benedict solution to give positive test. In summary, iodine solution detects the presence of starch. Benedict solution detects the presence of reducing sugars. Salivary amylase breaks down starch into reducing sugars, as demonstrated by a lack of reaction with iodine and the positive results with Benedict solution after enzyme activity. Question number 3. Role of the following in the experiment. Solution E, which we have seen is salivary amylase, breaks down or hydrolyzes or converts digestive or all those ones there. Remember the way we pick. You don't like all the ones there. The stroke there means alternative. accept glucose or simple sugars. Accept E breaks down D. Hot water provides optimum suitable or best or favorable or once to that effect temperature for effect of E for action of enzyme required when testing for reducing sugars using Benedict's solution. So that is the way we as examiners saw. Let's look at what I researched for E, salivary amylase. The role. Salivary amylase is an enzyme responsible for catalyzing the breakdown of starch into maltose, a simple sugar. It initiates the digestion of starch, breaking the long chains of carbohydrates into smaller molecules. Function in the experiment. In the experiment, salivary amylase is tested to demonstrate how it acts on starch. The enzyme's activity is observed by checking for the disappearance of starch using iodine solution and the appearance of reducing sugars using Benedict's solution. So, be keen on what I'm emphasizing, guys. Hot water bath, what I researched. A role of hot water bath is a hot water bath provides the heat required to activate the reactions, particularly for tests that involve Benedict solution. Function in the experiment. In the experiment, the hot water bath is used to heat the mixture when testing for reducing sugars with Benedict solution. Heating is necessary for Benedict solution to react with reducing sugars and produce a color change, indicating the presence of sugars such as maltose. Identity of E in humans is salivary amylase or thiarine, or you could have said amylase or pancreatic amylase. Reject one enzyme alone. D, or somebody saying E is an enzyme, you reject. D, explanation of the observations on the reagent tested with Benedict solution. Starch in D is non-reducing sugar or complex or polysaccharide, not a reducing sugar. Starch is hydrolyzed or digested or broken down to reducing sugar by E or by amylase or diastase enzyme in E. E must be mentioned. Reject amylase but accept amylase in E. Reject starch is a non-sugar because saccharides are sugars. Question 3. Here is an explanation of the observations made when reagents are tested with Benedict solution, which is used to detect reducing sugars like glucose, fructose, and maltose. So, before heating observation, the Benedict solution is typically blue in color. Explanation. Benedict solution contains copper 2 ions, which give it blue color. At this stage, no reaction has taken place. After heating, once the mixture is heated, observations depend on the presence and amount of reducing sugar. No change stays blue. Observation. The solution remains blue after heating. Explanation. This indicates that no reducing sugars are present. The copper 2 ions in Benedict solution have been reduced, so the solution remains unchanged. Green or yellow precipitate. Observation. The solution turns green or yellow after heating. Explanation. A small amount of reducing sugar is present. The reducing sugars reduce the copper 2 ions to copper 1 oxide, which is yellow or green in low concentration. Orange or red precipitate. The solution turns orange or red after heating. Explanation. A moderate to high amount of reducing sugar is present. The orange or red precipitate of the copper 1 oxide indicates a significant presence of reducing sugars. Brick red precipitate. Observation. The solution forms brick red precipitates after heating, although in Kenya we don't allow this. We don't allow this brick red thing, but maybe in some countries they allow it. But for the sake of knowledge, if you use that one brick red precipitate, which is not around in our examinations in Kenya, the explanation is that it indicates a range amount of reducing sugars. The higher the concentration of reducing sugars, the more copper 1 oxide is formed, leading to this intense color change. Summary of the observations. Blue, no reducing sugars present. Green / yellow, low concentration of reducing sugars. Orange, red, moderate concentration of reducing sugars. Brick red, high concentration of reducing sugars. These observations provide a qualitative indication of the amount of reducing sugars in the tested. Note, B1 is tied to the whole table. B2 is independent. C is tied to B1 and D is tied to the whole table. So when something is tied, if you miss what is being tied, you don't get the answer. Acknowledgement. Special thanks to all my teammates during marking exercise of the KCSE Biology Paper 3, who in one way or the other participated in coming up with the final draft of the marking scheme during the modulation as senior examiners. I also give special thanks to ChatGPT, an AI language model developed by OpenAI for providing assistance with creating content for this video. Goodbye, guys, and see you in my last video. And this is KCSE 2016 Biology Paper 3. Welcome also to that paper. Please don't forget to subscribe to this channel because knowledge will continue flowing from this channel. There is no limit in my videos as far as biology practical is concerned. Goodbye and see you in my next video.